Good afternoon. Welcome to Brainstorms, UT Brainstorms. This is our 9th Brainstorms, and we're really glad to have you, so many people here on a hot Saturday with the UT baseball team playing just down the way here. So thank you for coming. And so I want to tell you a little bit about Brainstorms, and then we'll launch into our program. In a second, I'll tell you about the format. In case you, how many of you, is this your first time at Brainstorms? That's amazing, thank you. So this has been a surprise to us. We've always had, how many of you have been to, say, 5 or 6 or more? Yeah, so this is what we have. We have our regulars, and we have this constant stream of new people, so fantastic. So the ones who raised your hand the first time, Facebook, how many of you found out through Facebook? Yeah. How many of you have a friend who's come here before? Okay, so that's awesome. So, excellent. So before we start, this is always fun for us. By the way, I'm sorry, I'm Mike Mauk. I'm the chair of the Neuroscience Department at UT Austin. And happy to be doing the last one of the brainstorms of the season. So if you're a faculty member, these are fun for us to do, and we get all excited and we get caught up in everything, and we're thinking about saying the right things and being on time and all this, and so we often forget to thank the people we're supposed to thank. And I've been guilty of this, and so I want to start with some thank yous. So this is Alma and Alondra, who both work for the Department of Neuroscience, and you met them out there. They do everything, and I wanted to make sure this last time to give them a big thank you. So one of the great things about the Department of Neuroscience is we have great loyal people like Alondra and Alma. They're here on a Saturday. They're not paid extra to be here, and they're loyal and they work hard. And so I wanted to say, so Alma's probably off doing something, isn't she? Thank you, Alma. I wanted to acknowledge Laura and Ian and Chris Weatherly. They're also people who work with us in the Department of Neuroscience. And Laura and Chris are my right-hand people that I couldn't do much of anything about. In fact, Laura and I have a pact, is that if either one of us leaves our job, the other one will leave too. So if she quits, I'm not going to be chair anymore. If I step down as chair, she's quitting. So we're a good team. And I also want to acknowledge we've had some volunteers over this season. Christine, Nikki, and Navia, who's here today. So Christine and Nikki are UT undergraduates, neuroscience majors, and so they're off for the summer. And Navia is here she is, hi. So Vandergrift High School student who's volunteered a couple of times, and so we're going to say thank you to you guys as well. So let me tell you a little bit about Brainstorms. So this is an outreach program that was quite intentional that we put the word conversation in the title. So we intended this to be a two-way conversation, a way for us to share with people of Austin our knowledge of neuroscience and how relevant it is to us. It's a fascinating field. It's relevant to our daily lives, and so we want to share that way. We wanted to hear from non-scientists, so you'd be surprised how excited we get and how inspired we get doing these things and hearing your stories and hearing and answering your questions. We go back to our labs and we feel more relevant when we do things like this, and so we meant it both ways. We want to talk about ways that we can help. I've spoken with a few people. We can connect you with faculty members or people who have expertise in something that you might be interested in or a problem you're having, although we're not physicians. You have to remember that. And we're interested in talking to you about ways that you can help us. We've had volunteers and a variety of people help us make brainstorms happen. And so we still feel that way. And you'll see even today that the format of this is that I'll give a little primer about today's topic, which is brain damage. But then we'll spend the majority of the time question and answers. Now, We're deviating just a bit from our normal format. Normally, somebody like me would speak, we would take a 5-minute break. You have 3x5 cards, each of you. If you feel like writing down a question, we'll be grabbing them at the break. And normally we would have a panel of people, and the speaker and the panel would answer your questions. It's June. June's, if you're a professor at UT, June is vacation time, and so I'm the panel today as well. So you're stuck with me. But we'll do the same thing. We'll take some questions from the audience. I'll take some questions from the cards, and we'll do our best to answer as many questions as we can. Just keeping in mind one thing: I'm not a physician, and it's just not right for me to be giving any clinical advice or diagnosis. I diagnosed my mom's Parkinson's disease, and I retired. I'm done. That was my clinical feat right there. And then the other way that we're going to deviate is that since this is the last brainstorms for the 2017-18 season, I want to stop about 10 minutes early. We have brainstorms t-shirts now, which which we will be ready to sell for next season of brainstorms. But today we want to have some fun giving away a few of them and just have a little fun at the end. All right? So this was an experiment for us. We felt there was a need for this, and we thought people would come, but we weren't completely sure. And I was thinking back today to the first brainstorms last September, and we all had different worries. So I was worried that 12 people would show up. And Laura and Alma were worried that 1,500 people would show up. And so what we know now is that we've had some good times and bad times. Our biggest problem, if some of you have been to the previous ones, we've had some overflow seating, and we probably, if the fire marshal had been here, we may have violated a couple of rules. And so, but it's exceeded expectations, and I thank you for attending and for doing that. We estimated this morning, counting today, that we had a total attendance this season of about 1,920 people, over 9 of them. So thank you very much for that. So, you probably saw the save the date. So we just signed up for 9 dates next year. About once a month, we're doing the same thing as before, sometimes on Saturday afternoons, sometimes on Thursday evenings. But some of you know this, but the big surprise is, so anyway, so here were the ones that we've done, the first 8, and so, but the big surprise then is that we made this was a goal of mine, and so we've done it. We made the leap, and we'll be moving brainstorms across the way behind me to the Lady Bird Johnson Auditorium, just a layer, 967 seats. So hopefully we won't be having any overflow. But as you can see, it's a beautiful auditorium. This would be your view, and this would be my view here looking out. It's a beautiful auditorium, much more space, and so we're hoping then to be more aggressive with advertising. We're counting on you to spread the word, tell people, bring a friend next time. But we're hoping to have crowds more like 500 and 600 next year. So it's about 4 times more expensive over there, and so we're a little worried about that. But the good news is that if you know a horn raiser, how many of you know that? So UT has its own Kickstarter thing called Horn Raiser, and they sponsor 2 or 3 of them at a time. We applied, and we were accepted for a horn raiser that's going to run September. And our goal is to raise about between $18,000 and $22,000. That's our goal, because that's the difference in cost between hosting it here and hosting it across the way. And so we feel like that so many people have come now, and there's a little more interest that we're just going to take the leap and gamble a little bit and have fun in the bigger auditorium next year. All right, so that's done with the commercial. So today's topic is the broken brain. And so I want to talk a little bit about how neuroscientists make use of the symptoms and problems associated with brain damage to help us understand how the brain works. And of course, it's an odd thing, really, in a way, trying to understand something by breaking it and seeing what happens. But we do this all the time. If you think about a car mechanic, I mean, you know how a fuel pump works, because when it breaks, you know that the engine stops because there's no fuel getting there. And one of the things we'll see as we talk is sometimes things are straightforward. Sometimes the symptoms of a particular neurological problem or brain damage are quite informative and similar to the way we would think about the function of that part of the brain. Sometimes it's really a bit of a misdirection. And we have to use other techniques. And so even though we're going to be talking about brain damage, I just wanted to give a whirlwind tour of the basic techniques that neuroscientists use to study the brain, just to remind us that we would never just stop at, well, it broke and this is what happens, and so that's our understanding. We always augment that kind of work with other things. And so here's my favorite cartoon of the brain. And of course, we already sort of know intuitively. We all know people who've had strokes and such. Everybody's familiar with and comfortable with the idea that different parts of the brain do different things. And so we're going to use brain damage and symptoms of brain damage to try to push that idea further. But let me just talk a little bit about the other techniques. So one technique that used to be considered old-fashioned and now is getting a modern revival is anatomy. So these are beautiful old drawings from Santiago Ramón y Cajal, a famous neuroanatomist in the 1800s. And what he had was this technique that stains neurons, just a small percentage of them so he could see them. And he was a fantastic artist. So these are hand-drawn pictures of different neurons from different parts of the brain. This is just him looking through the microscope at a slice of brain tissue that's been stained in this special way. And so you can see then that this is the hippocampus. We're going to talk about it in a minute. So you can see that hippocampus has neurons in layers, and different layers are doing different things. Here's the cerebellum, which has different kinds of neurons with different shapes. So the purpose of anatomy is to find out what kind of neurons are there and to understand how they wire together. So really the ultimate goal of anatomy is that it gives us a wiring diagram of different parts of the brain. And if that seems like an odd thing to hear, the brain is kind of an electrical device that each neuron generates signals and passes along those signals to other neurons. And so just like an electrical engineer would need to know the wiring diagram of the device to try to understand it, neuroscientists do much better at understanding how things work when we have a wiring diagram of the parts of the brain. I studied the cerebellum, and one of the reasons I committed to that about 30 years ago is that at that time the cerebellum was the best understood part of the brain in terms of the wiring diagram. It meant that we could ask more sophisticated questions and think about it more deeply, that our experiments could be more concrete. Now, over those 30 years, other parts of the brain have caught up, and we have good wiring diagrams of a lot of the brain. But that's the purpose of anatomy. This is a sort of a modern view. This is an amazing technique. This is called brainbow. So this is I know. The scientists just aren't good at it. This is an amazing technique. It uses a genetics trick to randomly turn different neurons different colors. So this isn't some sort of pseudo color image. This is a real image through the microscope. This is the cerebellum we were just looking at. This is the hippocampus. And it's a great technique to help us sort of untangle things. If we stained these all the same color, they would just be blobs. But stained like this, we can see their shape and what they're doing, and the red one's going over here and the green one's going over there. There are other beautiful genetic techniques like this that are really revolutionizing what we can do with anatomy. So then the other big one is that neurons like to record the activity of neurons and to see what's going on, because after all, that's what they're doing, and as they're processing information. So one of the powerful ways is to use functional magnetic imaging. So this is a picture of about a 3 Tesla magnet So this is one of these things, if you walked into the room with your car keys, that would go flying out of your pocket and smash there, and then you would be a whole bunch of trouble because you just broke their $7 million toy. But what people can do with these magnets is they can use a clever trick, which basically measures the amount of oxygenated blood in about a 0.5 millimeter resolution. And as it turns out, it's a real simple principle. The busier some part of your brain is, the more oxygen that's pulling out of the blood. And so this technique can find regions where the oxygen in the blood is lower. That means that part of the brain is active, or higher, it means it's not active. And so, for example, this is the kind of image you get. This is a snapshot of a living human, where the yellow parts have labeled particularly active parts of the brain in the test. Now, this experiment was done by a friend of mine, and it was literally about showing the effects of advertising on our brain. This was an experiment showing that Coke is particularly good advertisers, because the pleasure parts of their brain light up more when the people know it's Coke than when they're drinking an anonymous, unidentified cola. Okay, so it's the same sip, right? But when they know it's Coke, then these yellow places are brighter, meaning it's more rewarding. And so this is kind of scary. This shows that advertising affects our brains. So just a tad scary. Now, at a more fine-grained level, then, a lot of what goes on in research labs across the country is we can record the electrical activity of single neurons. And so this is the example of the kind of pulsatile activity that neurons make. It's these spikes in voltage that actually send signals to other neurons. And so this is an active neuron, and now it's quiet, and now it's active. And so all across the country, including here at UT, people are recording neurons like this and asking, when is it active, what's the mechanisms of active, when does it change during learning, etc. So then the other one that it's quite powerful, but I think lay people, non-neuroscientists don't know as much about, is stimulation. So it's possible, those signals are real, that's real electricity. It's not a metaphor that neurons make electrical signals. It's truly, current flowing and such. And because of that, then, there are relatively simple ways that we can implant electrodes in animals and pass the right kind of current, and we can force those neurons to be active artificially by passing the current. And some amazing things happen. So, for example, this happens sometimes. This is even done in humans, awake humans. So it's not uncommon if somebody has epilepsy and they're going in to do an exploratory surgery to find out exactly where the focus of the epilepsy is. They'll stimulate around there, and people will experience things. So as an example, we're going to talk in a bit about how this medial temporal lobe right here mediates certain kinds of memories. And if the neurosurgeon was stimulating there, people would report, oh, I just remembered that tree in our front yard when I was 7 years old. Okay, so it forced the person, the stimulation forced the person to have that memory by causing the neurons to be active. That's a quite powerful tool that is used in the lab a lot. But what we're going to talk about today is brain damage, because it's crude and sometimes it's more useful than others. But it's relatively easy for us to talk about, and we can wrap part of the pun, wrap our heads around. I love that phrase, but as a neuroscientist, it's not. So let's start talking about different kinds of damage. So this is a cartoon of a human brain, where the different lobes of the brain are labeled. So this sulcus right here is called the central sulcus, and it defines everything in front of it is the frontal lobe, and this is prefrontal cortex. Primary motor cortex is right here. So for example, a stroke that damaged here would leave somebody paralyzed. If you'd seen somebody with kind of a withered hand after a stroke, the hand region of primary motor cortex. The yellow is the parietal lobe. We'll talk about how that's an interesting sensory area. Occipital lobe in the back, that's really the seat of vision. You'd be surprised how much of the human brain is occupied doing vision. That's why we're such visual creatures, is we have a lot of our brain devoted to it. Temporal lobes for memory and visual processing, a few things like that. And somebody pointed out earlier, they complained that the cerebellum is in gray here. This is the part of the brain that I study. I meant to tell you, in a lot of pictures like this, they don't even bother with the cerebellum. So, yeah, because I study the cerebellum, I found one where that was not true. So as it turns out, let's talk about the cerebellum first. Cerebellum, it has very obvious symptoms when it's damaged, because those symptoms are motor. And so let me show you a movie of a woman who's had a stroke, a recent stroke, and it's a severe cerebellar problem. So she's not drunk, and you can see that she's not paralyzed, but you can see that she's badly impaired in the sense that all of her movements are inaccurate. You notice that everything she does is a little too big, or just not the right amount. So she's going to try to grab for this. She misses twice. So she's not paralyzed, but she's uncoordinated. And so we know for a long time, because of symptoms like this and because of the anatomy of the cerebellum, that for the longest time people talked about it as being important for sensory motor integration, which I have to tell you means absolutely nothing. Right? That's just a very generic term. What we know now from other experiments with stimulating and recording and anatomy and such is what the cerebellum does is that it uses learning to improve movements. So if you're familiar with the phrase muscle memory, athletes like to tell that sort of automatic sense that you've practiced something for a long time and now it's easy and automatic and you don't think about it, that's the cerebellum. Okay. Now what's the problem with this woman now is that the rest of her motor system is fine. The rest of her motor system is sending commands. Let's walk, let's reach for that fence. But the cerebellum is gone. In her cerebellum, she had a lifetime of memories of reaching for things and missing, and reaching for things and doing better, and walking and setting her foot down in the right place. And now with that gone, she has no mechanism to make movements accurate. And so it's not sensory motor integration, it's accuracy. What's absent here, we know from other non-brain damage experiments, is accuracy. So the cerebellum is there to help you make your movements accurate. Okay? But, as a person who studies the cerebellum, I know everybody says, yeah, fine, whatever, but it's not the important part of the brain. And the reason for that then is that the part of the brain that, as non-neuroscientists, we care about the most is the cerebral cortex, because the cerebral cortex is where our mind arises. And so in humans, the cerebral cortex is folded up. It's really just a sheet that's about 2 millimeters thick. And if you unfolded it completely, it would be approximately 1 meter square. And the reason why it's all wrinkly like that is it's folded up so that it can fit better inside of our skulls. And so we all know that different parts of the cortex mediate different aspects of our experience in our minds. And I'm going to take you through a whirlwind tour. We're going to talk about the temporal lobe, the frontal lobe, parietal, and the occipital lobe. We're particularly going to talk about the occipital lobe because even though the symptoms are less bizarre, I think we can use vision as a way to understand the most deeply the main point of today, which is even though we experience our minds as kind of a unitary thing, you're just you. Right now I'm here giving this talk, but what's happening in our brains is surprisingly disjointed. That different parts of our brains are calculating different aspects of our minds, and we don't know how all that comes together. It may be an illusion that it seems so uniform and it's like, well, I'm just one person. But what we'll see is inside of our heads, different aspects of your mind are going on in different places, and that's why brain damage can have such unusual and specific consequences. So that's what we're going to be talking about. So let's talk about the temporal lobe. In fact, we're going to talk twice about the temporal lobe. This is the inferior temporal lobe, this later. I'm going to be talking about the medial temporal lobe, which turns out to be important for memory. And this guy, Henry Molaison, was a great gift. His misfortune was a great gift to neuroscience. We learned so much about memory in the medial temporal lobe from him. So his story is that as a teenager, he had a bicycle accident. Wear your helmets. One consequence of that is he had terrible epilepsy and severe seizures, 7, 8, 10, a dozen a day. Drugs weren't working completely. Yep.
Did he lose his hippocampus?
What's that?
Didn't he lose his hippocampus?
Have you been reading ahead? Yes, he did. He didn't lose it, he had it taken out.
Yeah, so it has damaged it and he was using.
So they mapped his brain. So they said basically we can remove the trigger zone and get rid of your epilepsy, but it may cause you a little problem with memory. This is what they told him. Now, what happened is people, so his trigger zone was in this medial temporal lobe, and that surgery indeed had been done before. But HM or Henry Molaison was different in the sense is that part of what made his epilepsy so severe is that he had the trigger zone on both sides. And so they took out the medial temporal lobe on both sides. And realized almost right away that that was a big mistake. Because from that point on, and this is often misstated, so I want to be very clear the way I say this. From that point on, he formed no new long-term memories. People often call this a short-term memory deficit. In fact, all he had was short-term memory. So what does that look like? Okay, so after his surgery, you could say hi to him and then leave and come back 5 minutes later. Let's say that we met for the first time. Hi, how are you doing? 5 minutes later, come back. He has no memory of that because he can't form new memories. All of his old memories are intact. He was very good at recounting his childhood. All of the memories formed before his surgery were there. But he couldn't stamp in any new long-term memories. So he could remember something for a few minutes, but beyond that, then it never had, to his mind, it never happened. And this woman, Brenda Milner, was a neuropsychologist who got quite famous studying him. She made some fundamentally important discoveries about how even our memories are disjointed. We have different parts of the brain for different kinds of memories. So he had a very severe deficit in what we call declarative memories. It's the memories we're most familiar with, like what happened this morning? Did I meet you 10 minutes ago or not? Okay? But a lot of his other memories were entirely intact, and one of my favorite stories is this puzzle. This is the Tower of Hanoi puzzle. So this is a game where you can move the rings around, and you have to get this stack over here, moving one at a time, and you can never stack a larger ring on top of a smaller ring. Now this is the seven circle version, which means the optimal solution is 64 moves. So they would give this to Henry Molaison, and have you ever seen this before? No, he would play it, he was bad at it. They did it over and over and over. Every day he would report, yeah, I've never done this before. But over the course of 6 months, they would give it to him, and he would do it in exactly 64 moves. So he had a memory. He learned this game. He was really quite good. In fact, he would joke apparently, I'm pretty good at this, you know. But he had no memory of ever having played the game. And if you played it and did it in 64 moves and you went away and came back 5 minutes later, he would say, yeah, I've never done this before. Okay? So there are different, memory is not unitary. We have different kinds of memory, and with those different kinds of memory are mediated by different parts of the brain. As it turns out, the hippocampus, which you brought up, was the important thing that was removed on both sides. We know from things like this that one of the purposes of the hippocampus is to stamp in new long-term memories. And that's why you could do that. So that's the temporal. I just threw this in. So this is my dad, who passed away about 5 years ago. He had Alzheimer's, and the hippocampus and medial temporal lobe is one of the first parts of the brain that starts to go south with Alzheimer's. And early on, this was about the time that I started telling my family that I think Dad has a problem. And the reason I said that is he was HM. He was Henry Molaison. He was starting to, he could tell you, everything that he knew from the past, but he was having trouble remembering what happened 10 minutes ago. And we finally had to move him here to Austin, and we had him in a nice place, and I had a lot of guilt about moving my dad from California and having an intervention and treating him like a child. So I would call him, when we first moved him here, I would call him and say, hey Dad, I'm gonna come by, we'll have a drink, all right? And I learned pretty quickly there was no point in calling, because I would call him and I would drive over there, knock on the door and say, Mike, what a surprise, I wasn't expecting you. So I just learned to show up, because he was gonna say that whether I called him before or not, right? So early signs of Alzheimer's disease, think of HM, think of this problem of, and as my dad's Alzheimer's got worse, the people talk about the fog moving backwards in time. Eventually, Alzheimer's starts erasing existing older memories, and in a way that we don't understand, it seems to erase them sort of recent memories first. And I knew my dad's history. He, was in the Navy as a teenager, and then he lived in Kansas and then Minnesota, and then he moved to California. And as he got worse, I would talk to him, and I could see, after about a year living in Austin, California went away. He didn't remember living in California. And when even at his worst, he could still remember, I don't know if any of the stories were true, but he could still tell stories of being in the Navy in the Korean War as a teenager. Those memories, for some reason, because they were so old, were the last to go. So that's something to look out for. If we think of HM, when you think about maybe early warning signs for Alzheimer's disease. So let's talk about the frontal lobe now. The frontal lobe is interesting because it's really important for things that we hold dear, and in terms of our self-control and reasoning and things like this. And one interesting thing about the frontal lobe is that, among other things, it seems to mediate our morality, in the sense that people with frontal lobe damage have trouble controlling themselves. And so the classics are, who hasn't heard of Phineas Gage? Okay, so what? You know about HM? Well, you have heard. Yeah, okay, come on. So yeah, it used to be that nobody knew, and now thanks to Radiolab and things like that. So I want to tell a story anyway. So Phineas Gage is early 20s, really responsible. He's working in, I think, New Hampshire or Vermont, one of the two, on a railroad crew. He's helping lay the track for the Transcontinental Railroad going east to west. And he's the foreman of his crew because he's so responsible. And apparently, and this is this iron rod that was the cause of his accident. So the way they did it apparently is they would drill into the granite, pour in gunpowder, put a fuse and then a rag, and then they would take this big iron rod and tamp it down, and then light it, run, and then they do their thing. Well, because he was the foreman, presumably, he was the easiest job was maybe to tamp down with the iron rod. Bad decision by him. So something went wrong. Maybe they forgot the rag, but he tamped down and it sparked, and it shot this iron rod out of the hole, through his cheek, behind his eye, and out the top of his head. In fact, in the 1980s, they used his skull in some modern 3D reconstruction techniques to reconstruct the path of the iron rod through. So he lost a good share of his frontal lobe. Okay, so here's he was still intelligent, had all of his memories, was articulate, could answer questions and do math and different things, but he could not control himself. So instead of being the responsible person that he used to be, he was impulsive, and these people, this impulsivity is often kind of an inappropriate sexual way, that he would make lewd comments and just basically unable to control himself. So much so that he was unemployable, he lost his job. He was obsessed with this thing. He traveled around with this iron rod. He eventually ended up in a sideshow in some circus somewhere, and then finally ended up getting bad epilepsy and passing away about 13 years later in California. Okay, but he was the first case where people realized that what we might lose mind-wise when there's damage to the prefrontal cortex is about self-control and morality and behaving properly. Again, another one of these phrases that means nothing. Neuroscientists say executive control, which means I don't know, but I had to say something. Okay, but you can see the symptoms of this absence of control. Somebody mentioned something about prison inmates. So A while ago, there was a study that showed that over 3/4 of people in prison for violent crimes have some sort of prefrontal cortex issue. Okay?
What percent?
Over 3/4. So they're violent criminals, at least in part, because the part of their brain that they use to control themselves isn't working quite right. And as it turns out, without that, we don't behave particularly. So what's interesting then is one of the other things about, this is the lesser known thing about Phineas Gage, but of course, this is the superior or orbital cortex, meaning just above the eyes, just above the orbits. And we know now this is an interesting part of the brain in the sense that it mediates our ability to make decisions. So we're going to get kind of weird and specific now, okay? So, and what's interesting then is that this deficit doesn't come from the inability to process the information or to understand the facts. That's clear. It's an inability to pull the trigger. It's that person who says, well, what do you want, which movie do you want to see? I don't know, okay? So take that and multiply it by about 10, and that's what we have here. Now there's a neurologist at the University of Iowa who spent a career studying people with this damage, and we can just read this together. This is a recap of one of his patients. So the patient had come for a visit, and the task was simply to pick the date for his next visit. Okay, and so it says, I suggested 2 alternative dates, both coming month and just a few days apart. The patient pulled out an appointment book and began consulting the calendar. The behavior that ensued, which was witnessed by several researchers, was remarkable. For the better part of a half hour, the patient enumerated reasons for and against each of the 2 dates. Previous engagements, proximity to other engagements, possible meteorological conditions, virtually anything that one could reasonably think about concerning a simple date. He then walked us through a tiresome cost-benefit analysis and endlessly outlined fruitless comparison of options and possible consequences. It took an enormous discipline to listen to all this without pounding on the table and demanding that it's done. Okay? So what's important about this quote is he was in possession of the facts, but he can't bring them, for reasons that we don't fully understand. When this part of your brain is damaged, you can't bring these facts together and pull the trigger and make a decision.
What part is that called?
Superior orbital cortex. Sometimes superior frontal orbital or superior, right above the eyes. Okay? So it turns out, the other symptom of damage there is bland affect, blunted emotions. And I have an attorney friend who's a former judge and now teaches mediation here at UT, who was fascinated by that because his take is that in his generation, mediators were taught to let people vent to get rid of their emotions because people make bad decisions. And he, for a variety of reasons, felt that wasn't true. And he was fascinated when he heard me talk about this. And what started out as a beer on my back porch with my neighbor, one of my favorite neighbors, turned into, I now give 2 lectures every semester in his classes, one in mediation and one in negotiation, where I talk about how we make decisions with emotion. Okay, and in fact, this thing that the mediators used to do is wrong. And here's the thing: advertisers, politicians, they don't know this now. When was the last time you saw an advertisement that wasn't tugging on your emotions? Bluebell Ice Cream doesn't enumerate how great their ice cream tastes. They have these commercials where they talk about the good old days and how nice it is just to have a bowl of ice cream. You know. So politicians, anybody who's trying to influence our decisions are tugging on our emotions now more than telling us about the facts. Why? Because it works. This is something that we're vulnerable to. Right? So, and in part through this superior orbital cortex. So I love talking about the parietal lobe. The parietal lobe in general is a place with sensory processing, a lot of processing about our body and somatic sensation and such. But there's a very odd symptom that happens when sort of right about here is damaged, because one of the objects in the world is ourself. And there's this odd set of symptoms. that come from damage in certain parts of the parietal lobe called hemi-neglect. So hemi because it usually happens on one side, strokes usually happen on one side. Neglect is the key thing. People lose possession of the idea that objects have a right or left side, depending on what part of which the left or right side of the brain's damaged. So for example, somebody with parietal neglect, you might ask them to draw a clock. In fact, this is a classic thing to do in the clinic. And so what's odd is that they get the circle right. So somehow, circles are still there. But they are not in possession of the cognitive understanding that objects, in this case, have a left side. So they know that clocks have 12 numbers, and so they do goofy things like they put all the numbers on the right side. Now, as it turns out, when it's in the wrong place, then this neglect can be about one's own body. And so a real poignant way to see this is to ask people to draw a self-portrait. Now, this is an artist named Ronald Brown. So he's famous for these charcoal portraits. And he had a stroke and he had parietal neglect, and they asked him to draw a self-portrait. And this isn't him doing some Picasso statement, right? This is his view of the world right now, which is he doesn't understand the idea that he has a right side. Right? And I love this example because if you think about it, what's more self-evident than I have a right side or that objects have a left and right side? And what we learn from this is that the reason that's self-evident is because you have a part of your brain that makes it so. It's self-evident because your parietal lobe processes and says, of course, I can tell you about the right side. The other parietal lobe says, I can tell you about the left side. And when you lose it, then as weird as it sounds, these people don't even get the idea of the left side of the clock or the right side of my body. And it can lead to very oddly comical things when it's about the body. People will deny that it's not that they're blind, they just ignore one side of their body. You smack them on the arm and they'll yell, ow. And you'll say, what happened? They say, well, I don't know, there was just this pain. They don't say, you hit my hand with a hammer, because that doesn't exist, right? And so they just know that they felt pain, but have no idea. It's just this mystery pain that came out of nowhere. Oliver Sacks has some great, so if you've read Oliver Sacks' books, he has some great stories. One of his early books, the essay was called The Man Who Fell Out of Bed. So this woman took her elderly husband to the neurologist because he kept falling out of bed. Turns out he had a stroke, he had parietal neglect that involved his own body. He would wake up in the middle of the night and encounter his own right arm. And because he had this neglect, the only plausible explanation that he could come up with for the presence of his right arm is that somebody had played a horrible practical joke on him and put a severed, cadaver's right arm. And so he would grab it and try to throw it out of bed, and he would literally throw himself out of bed. Okay? It sounds comical, but he didn't have the part of the brain that told him that he had a right side. And so in the absence of that, that was the most sensible sounding thing he could come up with. Okay? And if you like things like this, the old Oliver Sacks book are full of, basically, I told somebody this morning that my lecture was just sort of Oliver Sacks live. So if you like this kind of thing, go grab some of his old books, they're really special. Okay, I want to finish with the visual system and the occipital lobe, because what we're going to find is so there's going to be visual problems. But this is where I really want to try to bring these ideas home that, again, we experience, think right now of what you're experiencing. It's like a movie. It's like a TV screen in front of you right now, right? It's a unitary thing, okay? But what we're about to learn from brain damage is that different aspects of your visual world are being calculated in different parts of your brain. And we don't know how it all comes together, but it does. And so here's what we see. Here are some specific deficits or damage to different parts of the visual system. So achromatopsia is an obvious and easy one, color blindness. That usually happens because of defective photopigments in the retina. But I'll show you in a second, there's a part of the cortex that when damaged, people just don't see color. Okay? Visual agnosia is an unusual one. It's quite severe. People can see, And they can report to you aspects of what they're seeing, but they can't bring those aspects together to talk about or to perceive objects. So it's a specific deficit in the ability to recognize and perceive objects. So you show somebody a picture of a horse, and they say, well, I see some brown vertical things, and I see, so they'll tell you about all the pieces, but they can't bring those pieces together and say, of course, that's a horse. Next picture. Okay. Then the flip side of this is prosopagnosia, which is similar. We have a dedicated part of our visual system that is used to process faces. We have no idea how good we are at processing faces. Our faces aren't really all that different, and yet in an instant we can look at each other and we all look different, right? We do that because, and it seems natural to do that because we have a part of the visual system that does only that. And when it's damaged, people can recognize objects, but not faces. And people with visual agnosia, they can't recognize objects, but they can recognize faces. Again, so it seems like your face is just an object in this room, but to my visual system, your face is being processed over here, and the objects are being processed somewhere else. And then the last one, which is really interesting, because it's another one of these self-evident, seemingly self-evident things. There's a part of our visual system that's important for processing the fact that things are moving. Akinetopsia, the inability to perceive motion. Again, seemingly self-evident. How you would think that the way we see motion, as I was looking, and then I looked again and it was not in the same spot. Well, as it turns out, that's not how our brains do it. We have a particular part of our brain that is there to report to us and calculate that objects are moving. I may have a problem with that part of my brain because I can't hit curveballs anymore in my stupid mental, and so I'm a little weird out there. But let me give you a tour of this. So the occipital lobe in the back here, that's the home of what's called primary visual cortex. It's the first place that visual information arrives. Second place, it goes to a part of thalamus, and then the retina to thalamus, and then thalamus to visual cortex. Damage there, you're just blind. You just can't see. Because this primary visual cortex feeds all of the other places. But so, for example, what was the first one? Achromatopsia. So there's a place about right here called V4, visual 4. Damage right here would make you colorblind. And not in the way that most people are with, but you wouldn't be able to discriminate different colors. Visual agnosia, the inability to identify objects. There are sort of two pathways that emanate from primary visual cortex. There's the ventral or what pathway, and the dorsal or where pathway they're called. So what pathway is objects? So for example, damage down here in the inferior temporal lobe, down here, caused that visual agnosia, the inability to identify objects. If you go right around the corner, it's underneath there, so we'd have to turn the brain and look up underneath here. But underneath there, around the corner, is called the fusiform cortex. Damage there is the prosopagnosia. You can't identify faces. So you're processing objects down through this, and you're processing faces just around the corner underneath. And then this region, so this is in now the where pathway, or right around here is called MT. And the MT region is the motion processing region. Damage there is what causes akinetopsia, the inability to notice that things are moving.
These things that you're discussing are focal vision. Do you do any work or talk about ambient vision?
I don't know what you mean by ambient vision.
Ambient vision is not actually seeing. Ambient vision is the way that your brain communicates with your eyes about what's going on around you. For instance, if you get car sick, that's your brain not able to process movement.
You know, I get motion sickness, and as a neuroscientist, I can tell you, we don't know why people get motion sickness.
Well,
It's really, well,
yeah, that's why I'm bringing it up. I have no idea what ambient vision was. I've never heard of it, but it's about 20% of your visual function is ambient.
Well, I think whoever's telling you that is talking about how a big part of how we see is that we have to control our eyes. So our whole visual world is messed up if we can't point our eyes at things.
Yes.
So if our eyes are bouncing around randomly, then it really disrupts our vision. And that will indeed make you sick.
Or if they're not at the same place at the same time.
Yeah, that's vergence. So if they're not pointing at the same place, that's a big mess too. That's right. Okay. So again, I saved vision for last because I just want to reinforce this point one more time. So you experience this one unitary movie, but different aspects, that movie is divided up into pieces and components, and those different components are being processed in different parts of your visual system. And this is why damage in these different places can produce specific and kind of odd deficits. What's it like to not be able to tell something that's moving, right? People struggle with this one. Like, it's hard to imagine. It's easy to imagine what achinotopsia or achromatopsia is, a color. You know, we have black and white televisions. We can kind of bond with that, right? What's it like to not be able to tell things are moving? Like, well, didn't you just notice that, well, it's not where it used to be? Well, as it turns out, if that's all you can do, you're really bad at noticing movement. So for example, people with damage to MT, it's really dangerous for them to walk around city streets because they look down the street and they see a car. And they don't know if it's a parked car, if it's barreling at them at 35 miles an hour. It's really dangerous for them. And if they look twice, they have to think for a second, wait, is that closer, right? And so this idea that we have that we notice movement because it's just not where it used to be, that's a really primitive and not very effective way to know. We have very elegant and complicated ways that we notice things are moving, and that's what this MT region does.
These 4 things, are they caused by damage, or can one be born that way?
Usually damage, yeah, stroke, gunshot wound, car wreck, tumor. Yeah.
What about concussion?
Yeah, concussion is its own crazy thing, because the thing about concussion is that it tends to be a little broader, because usually in concussion what happens is, we'll just divert for a second. The axons, the wires that neurons use to go communicate with others, they don't like to be stretched. And when you stretch them, it sets in motion a series of things that might make that neuron die 2 months from now. And that's really the insidious and bad thing about concussions. What happens is things get stretched, and you're fine now. But then 2 months later, you start having problems because that stuff got set in motion and now it's happening. And that's really the dangerous thing about it. And it makes it kind of widespread because you have all these axons from all these different places. They get stretched, and now you have all of these different problems. So concussion's bad.
And color blindness, I mean, I always thought you were born with that, but that's
By far the most common kinds of color, she thought that most people were born with color blindness, and that's true. The most common forms of color blindness is genetic defects where you don't have all of the color pigments in your cones in your retina. And in fact, there's one particular one that only happens in males that's the most common. It produces red-green color blindness. This would be a whole different way to be colorblind, which is you're fine, you have a stroke, and suddenly I can only see in black and white.
I recently dabbled in a lot of machine learning and regression analysis to try to figure out, differentiate things. And in the computer science area, we're trying to impose kind of the terminology from neuroscience into, just probability and statistics. And yet, when we approach, seems like the neuroscience area is approached with such a structured model that, even back to your chronology heads or this happens here and all of that. How does the brain essentially take unknown experiences, like experience of air space versus human space, and partition that out into I'm looking at foot air versus looking at human face, and make those distinguished fundamental buckets? So
we could digress into a long discussion because what's happening in all of these regions is processing. And not metaphorically, but in the way that a computer scientist would mean it. These different parts of the brain have different wiring diagrams that are specialized for whatever it is they're doing. And I can't answer your question about how you learned to distinguish a bear's face from a human's face, but I can tell you that it's processing of a certain kind in certain regions. Right? Now, I have to say, just that computer simulations are an increasingly big part of modern neuroscience. So for example, my lab has been doing computer simulations of the cerebellum for about 25 years now. So we build these simulations with all of our understanding of the pieces of the cerebellum, and the purpose of the simulation to ask, do we know enough of the pieces to explain the whole? And that can happen because it's really natural to use computer simulations to study the brain because computers compute and we're studying computation. So it's really pretty straightforward to build computational models of parts of the brain because that's what computers are good at mimicking, right? And so, okay, so let me just finish up then. So, not only do different parts of the brain do different things, but in a much more fine-grained way that you would ever imagine. That your mind is made up of all these different pieces that somehow come together. Now, once philosophers heard about this, they invented this term called the binding problem. And the binding problem is, wait a minute, you're telling me that all these different parts of my mind are being calculated into different parts of your brain. How does it come together? Okay? And I can tell you that most neuroscientists will say, well, look, if we're already going to have to accept the fact that all this neural activity in these neurons gives rise to my mind, why are we going to fuss over a detail like why is it happening in different places? The fact that this activity can give rise to our mind and our consciousness is enough of a miracle that we're already in miracle land. So most neuroscientists don't really think that there is a problem with the binding problem, but you can read a lot of neurophilosophy where people go on for pages and pages about the binding problem. How does it all come together? Okay. So what I want to do now is come together in this way. So we'll take a little break. One of the purposes of the break is for if you've written down questions, we're going to collect them quickly. And then I'm going to do my best to answer questions. You raise your hand and questions from the card. And then we're going to stop a few minutes early to give away some blank. Okay? So maybe just take 5 minutes and we'll get going. Thank you.